So next speaker is Alisa Piekny from University of Concordia University in Montreal.
Hi, so thank you for the organizers. Today I'm going to tell you about morphogenesis of the neuroblasts during embryogenesis. One thing that I've been really interested in throughout my career actually is morphogenesis in general. A common theme that has been emerging in the field lately is that you have these intermediate structures called rosettes. Let's see if my pointer works here. Rosettes that can form, and these are sort of, you can think of these as cell rearrangements. They're a combination of cell shape changes. And these rosettes are called because they're a flower-like organization, and you'll see that many of the cells will share a common vertex. And in some tissues, this could be a way to help elongate the tissue. So you can see that, for example, you have cells that would share, have a neighbor, would share a junction with that neighbor. They would disassemble. They would then go through cell shape changes, form this rosette, and then they would reorganize in a different axis. Now, in other tissues, this could be a way to help change or model the shape of the tissue. So again, you could see how apical constrictions, for example, in the subset of these cells, forming the rosette is causing the neural tube to help form. So there's different ways that rosettes can form. There's planar polarization that can occur. And I'm just showing you one, which is apical constriction. This is when you have actin myosin that's enriched at the apical edge of the cell. And these are, of course, these cells would be coordinated through junctions. And through these constrictions at the apical surface, they would create this sort of teardrop shape. And then you would see, again, if you took a top view, you would see this flower or rosette-like organization. Now, just to transition a bit, I'm going to introduce you a little bit to epidermal morphogenesis in C. elegans, which is what I had been studying initially. And I think Martha did a great job of introducing this already, but just to remind you, what I'm really interested in is ventral enclosure. This is when you have a subset of ventral epidermal cells that will migrate to cover the ventral surface of the embryo. And it's a two-step process where you have these two pairs of leading cells that will migrate first and meet at the midline, and then this is followed by the closure of a ventral pocket. And you can see that the cells in the ventral pocket will have these sort of constrictions toward their junction-free edges. And when ventral enclosure was actually first studied, and this really nice paper by Williams-Masson et al. Used phalloidin staining to show the organization of F-actin during this process, and you can see that this F-actin is highly enriched along the boundaries of these cells. And it was proposed at that time that these cells could be closing through actin myosin contractility. And subsequent studies from Pohl et al. Showed that through myosin localization, not surprisingly, you can see myosin nicely enriched along the boundaries of these cells where you would see, obviously, the F-actin. And at this time, you can almost imagine that a subset of these cells are forming sort of part of a rosette as they're kind of coming together to help sort of a coordinated constriction of the cells on the ventral surface. Now, something else that's really interesting about ventral enclosure is it really requires communication from another tissue, and that's the neuroblasts. So here. So they're drawn as these sort of green and blue circles in this figure, but what the Chisholm lab found back in the late 1990s and early 2000s is that there is a lot of signaling that's occurring to help these neuroblasts to independently sort. And this sortment is actually really important for ventral enclosure. And then the Soto lab found later that there are, of course, many of these axon guidance cues that are required, and the receptors are required in the epidermal cells. And so it's likely that these ventral epidermal cells are reading the chemical cues in these underlying neuroblasts, and then that's helping them to migrate and close properly. Another study, sort of more recently, by Ikegami et al., also showed that you have these maybe higher order organizations of the neuroblasts. So you can see, for example, a subset of the neuroblasts forming a bridge, and a couple of the posterior pairs of cells are actually using that bridge to migrate. So we actually became interested in a few questions. One was actually could there be mechanical cues in addition to the chemical cues that are coming from these neuroblasts to influence closure of the ventral epidermal cells? Secondly, what is the organization of these neuroblasts? There's obviously signaling that's occurring to create these unique patterns. So what is this pattern? Is this related to migration and closure of the overlying epidermal cells? And maybe the neuroblast tissue just has to undergo morphogenesis on its own, and this is an important part of embryogenesis. So one of the things we looked at was first to just show that myosin is actually required for ventral enclosure, because it actually hadn't been shown before. So we used a fast-acting TS allele, and indeed we saw that a significant proportion of embryos showed either delayed or rupture phenotypes consistent with ventral enclosure defects. And we also quantified this by just measuring changes in the surface area of the pocket during closure. And we could also see that not only is myosin required for this, but its upstream regulators are required for this. We also actually, though, were really more interested in whether or not myosin was required in the neuroblast. So we used a strain generated by the Chelfie lab, which actually is neuronal sensitive for RNAi. And we performed NMY2 RNAi in this strain and indeed saw that a significant proportion of the embryos had ventral enclosure phenotypes. So this was encouraging to us. And this sort of now prompted us to take a closer look at the organization of the neuroblast during ventral enclosure. And one of the things that we noticed was that as the overlying ventral epidermal cells came together to form this ventral pocket, and for that ventral pocket to close, we noticed that some of these cells were forming a flower-like pattern. And this pattern was somewhat reminiscent of what we had seen or what others had reported for morphogenesis of other tissues. And so we also then used seedwater segmentor software, which had been used in other organisms to track individual cells over time, and we indeed saw that those cells were sharing a common vertex when they appeared to show this rosette-like organization. We also used a fast-acting temperature-sensitive myosin allele to show that indeed the formation of this rosette was myosin-dependent. And what was interesting is that this upshift, we could still see cytokinesis occurring, which suggests that for some reason the thresholding of this allele is a little bit different for some activities versus others. But it also said that it's independent of, for example, cell division or other defects. So we also then wanted to look at myosin localization in the neuroblasts. And so what we did was we actually used a modified TIRF imaging. So we just shifted the plane of the angle of the objectives to image a little bit deeper into the embryo. And this actually allowed us to obtain a lot of information in a very fast, dynamic manner. So you can see these networks of myosin are highly dynamic within that pocket. So you can see here, and we see these foci that seem to sort of flow toward the ventral midline, and whereas foci in the head seem to remain a little more static at this time. And I'd like to point out that this allele was actually from, or this myosin probe that we're using is from the Goldstein lab. This is a CRISPR-generated NMY2. So we then wanted to correlate the localization of myosin with the rosette pattern that we had been seeing. Because if you notice, obviously the constriction of these cells to change shape and form this sort of rosette-like organization sort of involves obviously a myosin contractility, and myosin typically accumulates at the center of that vertex. So we just looked at the localization of myosin compared to the pattern of these cells, and when you see them starting to form this rosette-like organization, you start to see more stable accumulation of these myosin foci toward the center. Something else that was really interesting for us, and this is sort of a bit of a side segue, but we also wanted to know how perturbing the neuroblast could affect the overlying epidermal cells. Initially, we were just interested in the impact on myosin. And so if you perturb a neuroblast cell division using RNAi to NE1, we see that obviously the tissue is disorganized. Myosin is obviously no longer forming a nice pattern where you see central vertex, and there's obviously no rosettes. But we also saw this disorganized myosin in the overlying epidermal cells, which was really surprising to us because NE1 is not required for epidermal division. So we think that there could be some non-autonomous effect of the neuroblast tissue on the epidermal tissue, and we're currently investigating this. So something else that is really a common thing for rosettes is that they obviously, to undergo cell shape changes, you need to stabilize actin-myosin contractility through junctions. And so we then thought to start looking for the adhesion junctions in the neuroblasts. And in C. elegans, as Martha Soto pointed out, have several different components to the adhesion junctions. And so we looked both at the DLG-1, which is part of the DLG-1 AJM-1 component, and we looked at alpha-catenin, which is obviously part of the CCC component. And the alpha-catenin localized beautifully in the neuroblast, and we saw these central sort of accumulations that were highly reminiscent of what we had seen with myosin, whereas DLG-1 seems to be largely excluded from the neuroblast. So there could be some unique role for the CCC in the neuroblasts, and where the AJM-1 complex is not required. So we also looked at E-cadherin, and we're just now starting to really investigate the cadherin-catenin complex in more detail. So this is really new data at this point. But we noticed that the E-cadherin is also nicely expressed within the neuroblast, and we see it localizing to the cells that are coming together to form the rosette. But the pattern is a little more uniform compared to the alpha-catenin, which seems a little more punctate. And we also did RNAi to show that the E-cadherin is indeed required for changing the morphology of the neuroblasts during pocket closure, but it's not as strong as perturbing other components of myosin itself, for example. So this is something that we're still pursuing. Is there some redundancy, or maybe it's just that the RNAi isn't strong enough? Something else we've been doing is to try to characterize the rosettes in more detail. So we actually used a nuclear reporter, and we've been following the rosette formation using the nuclear reporter. And here we really could now beautifully isolate the cells that are coming together to form the rosette. And we noticed that there's 2 cells in the middle. And so what we think now is that that central focal point of myosin is actually corresponding to the top of those cells that are probably ingressing into the embryo. We also can see that the cells are really realigning into the different axes toward the end of ventral enclosure. So we do think that this is a process that's really helping that tissue to change its axis and shrink its surface area during ventral enclosure. We also see that some cells are migrating from quite far distances, whereas others are sort of more stably are migrating much shorter distances. And the ones that are migrating shorter distances are more part of the core, whereas others could be somewhat transitionary. So one cell could be part of the rosette, whereas another may not necessarily be. So I'm just quickly to say my model, we're just thinking of actually gastrulation as being a nice model for what could be happening with this rosette during neuroblast morphogenesis. We think that there's this coordinated myosin network. This is helping the 2 interior cells to actually ingress into the embryo. And then this network could also generate forces that could somehow be sensed by the overlying epidermal cells. And just quickly for future direction, we're looking at what cues could stimulate rosette formation. And we've really become interested in the can neuroblasts, which are actually migrating from quite far in the anterior to the mid-posterior of the embryo during this time. And one of the cans in particular takes a right turn and moves right into the center of the embryo, the position where the rosette then arises. So this is just a movie to show you that. So here's the cans coming in. One takes a turn. And now you're going to see the rosette cells come in and form that nice rosette. So we're now trying to investigate a role for the cans in this process. Is there a cue that they could be secreting, or is there a signaling pathway that they could be involved in? So I'm just going to wrap up. A quick summary that we now think we have a new sort of morphogenetic process that's occurring within the neuroblast to help that tissue develop with the embryo. We think that this is a myosin-mediated process, and I'd like to acknowledge the student that did most of this work, Denise Warnecka. Thank you.
So time for only 1 or 2 quick questions.
Sorry.
Hi, do you think this closure in C. elegans analogous to vertebrate neural tube closure in terms of the molecular machinery?
Yeah, so I'm not sure that I would necessarily model after neural tube closure. I think so right now it's really hard to tell the polarity of these cells. They haven't really been that well studied. So that's one of the first things we want to know and isolate. Is it apical constriction or is it actually planar polarity mechanism? That's something we have to first rule in or out. And then I think from there we would then go and try to model after other patterns and changes that occur in other tissues and see what molecules would likely be involved. But my guess is there's a lot of axon and guidance cues that could be involved in helping to initiate those cell movements.
Okay,
Yeah.
thank you very much, Alisa.